here to talk about. We have Dr. McDowell and also Dr. Wright over here to take in your data and your information and draw some conclusions about that. They'll be talking about that. But we're going to go ahead and get started. We had to make a couple of changes because we have some parents coming in to see presentations. There's a little bit of confusion about time. So hopefully everybody's got what's going on. The schedule that's up here is the schedule partially blocked by the computer. The way that schedule is what we're going to be working out of. So, the presentation up and ready to roll here.
I'm Maddox, this is Kendall, this is Christina, and this is Caitlin, and our project is on the effect of acetic acid on Arabidopsis. Many people, especially people in developing nations, rely on plants for their dietary iron supplement, which often results in iron deficiency. Iron's low solubility in LMIC diseases further add to widespread malnutrition. This experiment will test the effect of a lower pH via the addition of acetic acid on the wild-type experimental and unexperimental Arabidopsis thaliana. While no research could be found on the effect of acetic acid on Arabidopsis thaliana, studies have shown that A. thaliana grows best in a pH range of 5.5 to 6.5. The addition of acetic acid to other organisms has been found to increase iron uptake and stimulate stress response. The purpose of this experiment is to find a method of decreasing and ultimately eliminating world hunger through an increased iron uptake and disease resistance, and to test the effect of lowering the pH of a plant's growing environment, specifically Arabidopsis thaliana, on the agricultural efficiency of the plant's growth and reproduction. The null hypothesis predicted that the application of acetic acid would have no effect on either A. thaliana plant group. The alternative hypothesis predicted that the application of acetic acid would result in increased iron solubility and therefore increased iron uptake. This would then be reflected through the discoloration and stippling of experimental mutant A. thaliana plants. In total, there were 180 A. thaliana plants being tested. Half of these plants had an altered BTSG capable of 10 to 20% of its initial ability. Control groups were watered with 40 milliliters of distilled water, and experimental groups were watered with 40 milliliters of 0.018 moles acetic acid solution on Mondays, Wednesdays, and Fridays. Plants were exposed to 16 hours of light and 8 hours of dark using a timed artificial lighting system. Qualitative and quantitative observations and data were recorded on Wednesdays and Tuesdays and Thursdays. In order to reduce error, we had the same people complete each task throughout the duration of the experiment and use the same tools for measuring. Plants were watered and measured on the same days at approximately the same times. Despite our efforts to reduce error, experimental and control plant groups were placed in the same growing tray for the first 2 weeks of the experiment after watering, so this likely influenced the results of the experiment as control groups were likely exposed to acetic acid solution. This graph shows the comparison between all 4 Arabidopsis thaliana on average rosette widths. The wild types shared a similar growth pattern and had wider rosettes than the mutants, which also had their own similar growth pattern. This graph shows the comparison between the mutant plants, and there was shown to be little variance in average rosette widths. Next is the experimental, and in the beginning, they both shared a similar growth pattern. Yet on September 19th, the wild type experimental began to develop wider rosettes, and began to have wider rosettes, yet both began to grow at a similar rate. Again, this graph compares the controls, and just like the experimentals, they both shared a similar growth pattern in the beginning. Yet on September 19th, the wild type control began to develop wider rosettes, yet they still began to grow at similar rates. Next is the comparison on average bolt height. Just like the rosettes, the wild types shared a similar growth pattern and had higher bolts than the mutants. Although the mutant experimental began to develop bolts before the mutant control, they both shared similar growth patterns and kind of grew at the same rate. This graph compares both experimental Arabidopsis thaliana bolt heights. The mutant experimental began to show bolts before the wild type, and they grew at similar rates until mid to late October, when the wild type diverged from the mutant and began to show more taller bolts. This graph compares both control Arabidopsis bolt heights. They grew at similar rates once again until mid to late October, when the wild type began to show taller bolts. This graph compares the selene count in all 4 Arabidopsis plants. As you can see, they grew at very similar rates, with the only true outlier being the mutant control. Although the difference may look significant, this graph has large intervals, so the difference was only 1 or 2 seconds. This graph compares both mutant Arabidopsis selene counts, which also grew at the same rate, and also showed that the mutant control was somewhat of an outlier. This graph compares both experimental Arabidopsis selene counts, which once again grew at very similar rates, until mid to late October, when the wild type began showing a few more selenes. The final graph shows the comparison between both control Arabidopsis selene counts. Once again, these grew at the same rate until mid to late October, when the wild type began showing more selenes. Wild type control plants were overall taller and healthier looking than the mutant control plants, although they did still suffer from slight discoloration and shriveling of the rosettes. Mutant control plants were smaller and they had darker colors such as green, purple, blue in the rosettes. Both plants were very green from the bolt up, and they had bright green leaves. Wild type experimental plants suffered from discoloration. They turned pink and purple, and they were lighter than the mutant experimental plants. The mutant experimental plants were darker, and they suffered from more shriveling of the rosettes. Both plants had yellowing leaves, and the soil in the pots seemed dry and unhealthy. Mutant experimental plants were taller, however, they did suffer from more discoloration. They were less green, they showed more signs of pink and purple, and their leaves were much more shriveled and curled. The mutant control plants, however, still suffered slightly from this discoloration, potentially due to our mistakes earlier on in the experiment, but were shorter and healthier looking than the mutant experimental plants, although both plants had during all 3 growth phases of Arabidopsis thaliana in this experiment, the 4 plant groups had similar growth patterns. Qualitatively, the wild type experimental and mutant experimental plants were affected more than the wild type control and mutant. But quantitatively, the data had no significant differences between the plant groups. All of the plants were noticeably stressed and negatively affected. A surprising result was the amount of stress that the mutant control and wild type control plants were under, considering acetic acid was not intentionally added to their growing environment. An error that may have influenced our results was placing the control and experimental plants in the same growing tray after watering them, allowing for acetic acid to potentially seep from the experimental pots into the control pots. This error was noticed on October 2nd, and for the remainder of the experiment, the experimental and control pots were kept in separate growing trays. This error may account for the amount of stress the control plants were under. The data collected and results failed to reject the alternate hypothesis and rejected the null hypothesis. The alternate hypothesis states that the addition of acetic acid would have a greater negative impact on the mutant experimental because it would be unable to control iron uptake. While an increased iron uptake may account for the negative effects on the mutant experimental, the same negative effects were also present in mutant control, wild type experimental, and wild type control. The relatively similar data supports the idea that the addition of acetic acid had no significant effect on the growth patterns and overall health in the comparison of the plant groups. These results show that lowering the pH of a plant's growing environment to a level below optimal growth pH has no beneficial aspects to offer. Ultimately, this research can be used to eliminate the idea of growing plants in a stressfully low pH environment in order to increase iron uptake and quantity of crop yields. Although a lower pH has been found to increase iron uptake in other organisms, lowering the pH of a plant's growing environment to a stressful level has no benefit, hinders the plant's growth ability, stresses the plants, causes the plants to reproduce faster, and presumably produces a lower quantity and quality of crops. We now open up the floor to any questions.
So this isn't really a question so much as it is an observation. I really appreciate how you got some troubleshooting this here halfway through figuring out this problem, finding separation between experimental and control. What I really appreciate is how forthright you were about reporting this. That's how science should be done, right? We learn to work with our mistakes and we move on towards successes. And best practice is to share those mistakes as wrongly as possible. One of the reasons that it takes 5 years to get a PhD is that this sort of dynamic is really helpful. The first version of the experiment design can often be here. You have to go back and redesign, and you have to go back and redesign again after you figure out the second mistake and so on and so forth. So that's a really fundamental business. I think it's really good to acknowledge when you made mistakes and describe them clearly. So it's a great job.
Are there any more questions?
chosen as our independent variable, copper uptake is directly related to iron uptake. Copper inherently lowers iron uptake, which is related to the Brutus gene, which is 10 to 20% activity in the Arabidopsis plants. Copper sulfate was applied to both experimental groups in human and wild-type plants. 3 hypotheses were then formulated, comparing the other plants to the wild-type control. The first hypothesis, which compared mutant control to wild-type control, stated if the growth and health of the wild-type Arabidopsis thaliana plant in a control environment and Brutus mutant A thaliana plant exposed to excess copper sulfate are compared, Brutus mutant plant will follow in response to an excess iron uptake due to the decreased effectivity of the Brutus gene. Quantitatively, there will be a decreased rosette diameter, decreased pole height, fewer seed pods produced. Qualitatively, there will be absence of trichomes, presence of anthocyanin, and there will be deep growth. The second hypothesis, which compared the wild-type control to
wild type experimental state. If the growth and health of the wild type Arabidopsis plant in a control setting, wild type A plant exposed to excess copper sulfate or compared, plant exposed to excess copper sulfate will exhibit the following in response to lower oxidation. Quantitatively, there will be decreased rosette leaf diameter, decreased fold height, fewer seed pods produced. Qualitatively, there will be absence of trichomes, presence of anthocyanin, and there will be wilting in the leaves. Third, final hypothesis, which compared the wild type control to the mutant experimental state. If the growth and health of the wild type Arabidopsis plant and Brutus mutant A plant are compared in a constant environment, Brutus mutant plant will exhibit quantitatively there will be a decreased rosette leaf diameter, decreased fold height, fewer seed pods producing. Qualitatively, there will be absence of trichomes, presence of anthocyanin, and there will be leaf wilting. We kept our plants in the light box. The light box allowed us to keep the plants in a very controlled environment. It allowed them to grow in 6 weeks with sun, the second generation that already began to grow. Plants were kept in the light for 18 hours, in the dark for 6 hours. The temperature of the box was kept between 24 and 26 degrees Celsius, however. Arabidopsis plants generally like to be kept in degrees between 20 and 22 degrees Celsius. To counteract this, we opened the lid of the box to try and decrease the temperature. This may have been an unintentional variable. It may have influenced our results. When we first formulated our hypotheses, we thought the rapid growth was actually a sign of positive health in the Arabidopsis plants. But we later found out that the rapid growth is actually a sign that the Arabidopsis plants were under a great deal of stress.
One source of quantitative data collected to determine the overall health of the Arabidopsis plant is the rosette leaf diameter. About a week after being planted, the plant sprouted rosette leaves, which are typically the first pair of leaves to develop. As you can see on this graph, initially the rosette leaves grew very rapidly. Then, on September 27th, the 18th day of the experiment, the rosette seemed to steady out and grow at a slower rate. The decreased growth rate observed in the rosette indicates a shift in energy to prioritize the development of bulbs rather than the further growth of the rosettes. 6 days later, on October 3rd, the 24th day of the experiment, the plant sprouted bulbs. The development of bulbs is the next stage in the Arabidopsis reproductive cycle after the development of rosettes. This graph represents the same data but with removed dates. The removed dates allow specific trends and patterns to be focused on. It is important to note that 1 on the x-axis represents the beginning stages of rosette growth, and 6 on the x-axis represents the end stages of rosette growth. Stage 4 represents day 18, the first time a decreased growth rate is observed in the rosettes. Between stages 4 and 5, the experimental plants were given their first copper sulfate treatment, likely causing these plants great amounts of stress. The first hypothesis discussed a comparison of the wild-type control and mutant control plants. We initially hypothesized that mutant control plants would exhibit a decreased rosette diameter when compared to the wild-type control plants. As you can see on the graph, the mutant control plants, represented by the blue line, did exhibit a decreased rosette diameter when compared to the wild-type control plants, represented by the green line. This indicates a more rapid shift in energy by the mutant control plants to develop bulbs. In this case, mutant control plants were likely more stressed and less healthy than the wild-type control plants. The mutant control plants were likely more stressed and less healthy because they had an altered Brutus gene. The second hypothesis discussed a comparison of the wild-type control and wild-type experimental plants. We initially hypothesized the wild-type experimental plants would exhibit a decreased rosette diameter when compared to the wild-type control plants. As you can see on the graph, the wild-type experimental plants, represented by the purple line, did exhibit a decreased rosette diameter when compared to the wild-type control plants, represented by the green line. However, the decreased rosette diameter observed in the wild-type control plants occurs only after the 5th stage. This indicates a sudden shift of energy to develop bulbs. This was likely because the wild-type experimental plants were experiencing some sort of stress that the wild-type control plants were not. The source of stress for the wild-type experimental plants was most likely the excess copper sulfate they were being given. The third hypothesis discussed a comparison between the wild-type control and mutant experimental plants. We initially hypothesized mutant experimental plants would exhibit a decreased rosette diameter when compared to the wild-type control plants. As you can see on the graph, the mutant experimental plants, represented by the red line, did exhibit a decreased rosette diameter when compared to the wild-type control plants, represented by the green line. Following the trend mentioned earlier, the decreased rosette diameter observed in the mutant experimental plants occurs only after the 5th stage. Once again, this was likely caused by a sudden shift in energy to develop bulbs. The mutant experimental plants were also likely experiencing some sort of stress that the wild-type control plants were not. The source of stress for the mutant experimental
they not only had an altered British gene, but were also being given excess copper sulfate. A common sign of stress in plants is an accelerated life cycle. Typically, Arabidopsis plants do not develop bolts until about the 4th week of their experiment. In our experiment, all of the plants began to develop bolts at the end of the 2nd week. This is a sign of extreme stress.
Another form of quantitative data used in this experiment to determine the overall health of the Arabidopsis plants was bolt height. The bolt of the Arabidopsis plants is a stem that rises from the center of the rosette piece and is closely related to reproduction in these organisms. Following the 31st day of the experiment, all 4 groups of Arabidopsis plants showed an increasing rate of growth of bolt height. This is an indicator of stress in these plants as they were developing bolts at a much more rapid rate than a normal and healthy Arabidopsis plant. Additionally, the wild-type experimental plants, indicated by the color purple on this graph, were the first to begin bolting. Once again, this indicates that the wild-type experimental plants are under some form of stress because they prematurely developed their bolts. One source of stress for the wild-type experimental plants may have been iron deficiency. Not only did they have a fully functioning British gene, but they were also exposed to copper sulfate on a weekly basis. Therefore, both the British gene and the copper sulfate likely competed with the uptake of iron in these Arabidopsis plants, thus resulting in iron deficiency. You may also notice that between October 2nd and October 4th, there is a gap between the measurements of bolt height for the wild-type experimental plants. This is because on October 3rd, the group who had the plants that had begun bolting did not take measurements, thus resulting in a gap between the data on October 3rd. By the 40th and last day of the experiment, mutant control plants, represented by the color blue, had the greatest bolt height of all of the groups of plants in this experiment. They were followed by the mutant experimental plants, represented by the color red, who had the 2nd greatest bolt height of all the Arabidopsis plants. Wild-type experimental and wild-type control plants, indicated by colors purple and green, had 2 of the lowest bolt heights by the end of this experiment. This indicates that overall, the wild-type plants were more healthy than the mutant plants during this experiment. This graph has the same data, but without the specific dates that the bolt height was measured in order to allow us to focus more on patterns and trends in the data. Stage 1 represents when we first began measuring the bolt height, and stage 6 represents the last day we measured bolt height. As you can see, the mutant control plants, which are the color blue on this graph, had the greatest rate of growth of bolt height throughout the majority of this experiment. This indicates that the mutant control plants were likely the most stressed and least healthy plants during this experiment. One source of stress for the mutant control plants may be iron excess. Because the British gene in the mutant control plants was limited in function, these plants were likely to be taking up more iron than a normal healthy Arabidopsis plant. Though iron may be beneficial to plants, when in excess it can become toxic. Following the mutant control plants were the mutant experimental plants, indicated by the color red on this graph. They were also one of the more stressed plants during this experiment, likely because of nutrient excess. Not only did these plants have a British gene that was limited in function, they were also exposed to increased copper sulfate on a weekly basis. Therefore, they were taking up much more iron and much more copper sulfate than a normal Arabidopsis plant would, and nutrient levels in these plants likely became toxic. Wild-type experimental plants, indicated by the color purple, had a slightly greater rate of growth of bolt height than did the wild-type control plants, which are the color green. This indicates that the wild-type experimental plants were slightly more stressed than were the wild-type control plants. Overall, the wild-type control plants were the most healthy during this experiment because they had the lowest rate of growth of bolt height. This makes sense because the wild-type control plants were exposed to the most normal conditions of Arabidopsis plants. They were not given copper sulfate on a weekly basis, and they had a fully functioning British gene, meaning that they were not experiencing any form of nutrient excess. One pattern we noticed in our data was when you compare the rosette diameter to the bolt height of these plants, the 2 data are actually inverses of one another. For example, when looking at the mutant control plants, which are the color blue on these graphs, they had the greatest bolt height, but they also had the smallest rosette diameter. Similarly, the wild-type control plants, represented by the color green, had the lowest bolt height with the greatest rosette diameter. In addition, we also observed an inconsistent presence of capsules that grow off the bulb to allow the plant to reproduce. A higher seed pod count directly relates to a higher reproductive success rate. As you can see, we measured seed pods on 4 different days. As wild-type experimental plants were the first to exhibit seed pods, followed by the other plants 3 days later. However, all plants showed an accelerated growth rate, showing signs of the second generation within 6 weeks. The typical timeline for this is 8 weeks. Wild-type experimental plant, as indicated by the purple line on the graph, has the lowest seed pod count. This is because the fruit is used controlling the amount of iron to use the plant. Also, copper sulfate is controlling the iron to use. Due to this, not as much iron can reach the plant, decreasing the nutrients available for it to grow seed pods. This is why it has the lowest. Wild-type control plant has the second lowest, as indicated by the green line on the graph. This is because the pressures were decreasing this plant. It had an unaltered gene, and its conditions were left unaltered. This put no pressure on the plant to rush its growth and produce seed pods faster or at a higher rate. The mutant experimental plant, as indicated by the red line on the graph, has the second highest. This is because the Brutus gene did not control the iron uptake of the plant. However, copper sulfate did lower the amount, although it did have more nutrients than the wild-type experimental. Due to this, the nutrient excess could have been in the plant. This would have caused the plant to gain stress and reproduce quicker. The mutant control plant, as indicated by the blue line on the graph, had the highest seed pod production. This is because the Brutus gene was not controlling the iron uptake of the plant. Due to this, iron levels in the plant could have become toxic. This toxicity would have led to stress in the plant. The plant's normal response to stress is release of growth hormones. These hormones would allow the plant to grow faster, to reach reproductive stages, to reach its ultimate goal of reproduction. This is why the mutant control plant has the highest seed pod count. Our first hypothesis shows that the mutant control plant has the highest reproductive states out of wild-type control versus the mutant experimental. Our second hypothesis shows that the mutant wild-type control had a higher success rate than the wild-type experimental. And our third shows that the wild-type control was less than the mutant. Patterns between the bull height and the seed pod production are very similar. Both have the mutant control as the highest and the wild-type experimental as the lowest. This is because both organs are controlled by the reproductive system. Seed pods are the direct representation of the conditions found in plants, leading to different reproductive success.
Quantitatively, the mutant control plants were rejected because there was an increase in bulb height in the mutant control and more seed pods. Qualitatively, it was also rejected because there was a presence of trichomes and not an increase in bulb height in the mutant control. Overall, this whole hypothesis was rejected. Our second hypothesis was quantitatively supported because there was a decrease in bulb height in the wild-type experimental group and fewer seed pods. It was rejected qualitatively because there was a presence of trichomes and not an increase in anthocyanin in the experimental group. Our third hypothesis was quantitatively rejected because the bulb height was higher in the experimental groups and it had more seed pods. Qualitatively, it was accepted because there was more leaf wilting and a presence of anthocyanin in the experimental group. There were several errors in this experiment that could have caused inaccurate measurements. Yellowing in the leaves could have been overcrowding, lack of light in the plants, and copper sulfate being dropped onto the leaves. Copper sulfate is left unrefrigerated and uncovered, which allowed particles to enter the solution that was then added to our plants. Incorrect measuring was another error in this experiment because the bulb heights became too high to measure the calipers we used to ruler. Another error was that the overcrowding caused it to be harder to measure the rosettes. The data gathered in this experiment is of importance in increasing crop yield and world hunger. We also learned if the Brutus gene was beneficial or harmful to the Arabidopsis plants. Future research can include doing more trials of this experiment for more accurate results, or trying different nutrients that are linked to the uptake of iron. It can also thrive up under different environmental stressors. All of these would contribute results that would help us to figure out how to produce plants faster and more resistant to disease. We have figured out that the mutant gene and the plants with excess copper do grow and reproduce. They are under more stress, which causes their offspring to be of lower quality. They still reproduce, but do not have the benefits of the wild type control plants. British gene being suppressed is not beneficial because the plants still reproduce, but are not any better than the wild type control.
I would like to thank Virginia Tech and Dr. McDowell for this amazing opportunity. Thank you.
So first of all, I really like how you interpret results by tying together molecular biology of the greatest protein, plant physiology, and how that linked to it's impressive that students. Sorry. What was your feeling on the light box? Thumbs up or thumbs down?
I would overall give it a thumbs up. I think it created a very controlled environment and allowed our results to be as accurate as possible.
Thanks for the great presentation. I really liked the way that you marched some of your data down to better show the difference in things. Struck by some of your results showing how the treatment actually rescued this mutant, and wanted to hear about what follow-ups you might do to see if your hypothesis that iron excess causing that stress. How would you vary the iron concentration?
So in this experiment, we did predict that the mutant experimental plants were doing better than the mutant control plants because they were taking up more copper sulfate. And as Isaac mentioned in the introduction, copper sulfate competes with iron uptake. So in the future, if we wanted to verify that the mutant control plants were affected by excess uptake of iron, we could place the mutant control plants in environments containing different amounts of iron to see if that is what is resulting in rapid growth of the bolt height and seed pod production in the mutant control plant.
Take about a 10-second stand up.
Oh my god.
Okay, orange fourth, come on up. Everybody else, take your seats.
everyone. My name is Desire Alexander. These are my partners, Ryder Rose, Caitlin Shiflett, and Natalie Pettigen. And today we are presenting to you the effects of overcrowding on Arabidopsis thaliana plants. We are studying the mutant form of Arabidopsis thaliana because of its gene Brutus, which regulates iron uptake in Arabidopsis plants. The reason we chose overcrowding is because not only can it regulate iron uptake, but it also is immune to disease. We hypothesized that the wild type control will do the best out of all the other groups since it does not have the BTS gene and it is grown in normal conditions. We also hypothesized that the wild type experimental group will do the worst since it does not have the BTS gene and it is going to be presented to the overcrowding condition.
Our hypothesis for the mutant control group is that it would do worse than the wild type control group, but it would do better than both of the experimental groups. We also thought that their seed pod production would do worse than both of them. For our mutant experimental hypothesis, we thought that it would be the better of the two experimental groups, but it would do worse than both of the control groups. We also thought that for the seed pod reproduction, it would do the worst of both groups. The purpose of this experiment was to test the effects of overcrowding on the Arabidopsis thaliana plant with and without the mutation. Using the data that we collected, we can help prove that the Brutus gene can improve the agricultural survival and production of the plants that we need to survive.
For this experiment, we tested 4 groups in total, consisting of 2 experimental groups and 2 control groups. The wild type control group consisted of a total of 5 plants in total, with no BTS mutation, while the wild type experimental group consisted of a total of 9 plants with also no BTS mutation. Wild type control is expected to do the best, seeing as how it is we used it as a baseline for comparison. Mutant control consisted of a total of 9 plants, but mutant control consisted of a total of 5 plants with a BTS mutation, just like the mutant experimental, which consisted of a total of 9 plants with BTS mutation. Our variable was introduced at around day 7 of our experiment. Seeing as how we didn't have a chemical to introduce, we picked 2 numbers to thin our plants down to. 5 for our controlled and 9 for our experimental. The graph shown depicts the average rosette width for the groups. From day 8 to day 28, we measured our rosette's growth, and stopping at day 28, seeing as how bolts began to sprout, signifying the end of the growth. The wild type control did the best, with the wild type experimental not far behind. Meanwhile, both mutant groups kept growing at relatively the same rate and grew to be about relatively the same size. The image shows how the mutant groups were different in size and differentiated in color from the wild type group. The graph above shows the average bolt height of the Arabidopsis plants at phase 2. Since the beginning of the measurement, the green line, which is the wild type control, did the best, ending with an average height of 241.7 millimeters. Next was the mutant control, which had a rapid increase in growth until about day 33, when it had a steady growth, ending with an average height of 205.7 millimeters. The wild type and mutant experiments both were going back and forth throughout their growth cycle until the end on day 44, when the wild type control passed the mutant experiment with an average height of 205.3 millimeters, and finally the mutant experiment ended with an average height of 200.5 millimeters.
This is the graph of the seed pod reproduction phase. Throughout the entire experiment, the wild type control group did the best overall growth-wise, but in the end, the mutant control did the best seed pod reproduction-wise, a total of 35 seed pods per. On the other hand, as we suspected, the experimental plant did the worst in seed pod reproduction-wise, and includes a total of 22 seed pods per plant, which is still a fair number. Both wild type groups varied throughout the entire experiment. Whether it was in group 1, group 2, or group 3, they eventually each totaled out to about 27 to 28 seed pods per
In the end, the mutated Arabidopsis thaliana had a darker green color wild type. Also, the rosette widths were not as wide, and it also had a shorter full head. The seed pod count usually shows how well they lived in their environment. But although our data was incomplete because there was an inconsistent pattern. Wild type did grow better than the Brutus mutation plant, but the Brutus did withstand the stress of an overcrowded environment. For our experiment, it can be taken outside so we can see how a normal environment affects it. Also, we can add more plants to the experiment to see how much stress the bean can withstand. BTS1 mutation in our experiment, or Arabidopsis thaliana, stress did not grow as well as we expected, but still grew, which means there's hope for this mutation.
Yeah. So first a comment, plants look terrific. These are some of the best garden Arabidopsis I've seen. So it looked like there was an interesting difference with regard to rosette diameter crowded versus uncrowded. You know if those differences were statistically significant? Did you get that level of analysis in any way? So most likely not. It was striking that there was no difference in crowded and uncrowded plants for guys probably noticed that. Do you have an explanation for it? All right. Stuff. So you guys out in the audience are perfectly welcome to ask questions. It doesn't have to be just me and play. Some of the most fruitful exchanges that I've had in this program in the past are with the students involved. So don't be shy.
on how cold temperatures affect Arabidopsis thaliana with the inhibition of the Brutus gene. Our class was trying to see how Arabidopsis thaliana grows and reacts when the Brutus gene is not suppressing the BOS gene. As discussed by Mr. McDowell, the Brutus gene is responsible for regulating iron uptake. The BOS gene, which our experiment is centered around, is the gene that allows plant growth to thrive in cooler temperatures and drought. Our class was curious to see if the mutated Arabidopsis plant would be more successful growing in the colder temperatures because the Brutus gene is not able to suppress the BOS gene. Our class worked together to create the following hypothesis. The Brutus gene controls the BOS gene. The BOS gene controls the plant's reaction to drought and freezing temperatures. If we place the wild type and control, the wild type experimental and mutant type experimental in an environment with a cooler temperature, then the wild type plant will have a smaller rosette width, smaller bolt height, and will take longer for the bolt to appear than in the mutant type plant. This is an image of our plants on week 1 when we started our experiment. The only notable difference at this point is that the wild type plants are much thinner than the mutant plants. In order to test our hypothesis, we decided that we were going to place our plants in a refrigerator at 50 degrees Celsius for approximately 7 hours on Monday, Wednesday, and Friday. We also collected data on these days. This is an image of group 3's plants on week 6 as we concluded our experiment. There's many more notable differences compared to the previous picture. Although the wild type plants remain thinner than the mutant plants, the wild type plants had a larger average rosette width. The only bolt that we had for group 3 was in the wild type control.
After the 6 weeks of experimentation on these plants, we came back with results that went against our hypothesis. Initially, we thought that supplying the BTS gene and the BOS gene, which allows them to thrive in the cold, that the colder temperatures would better affect the BTS plants. But after the end of the experiment, the wild type control plants did the best, as you can see with the green line. In the end, both the experimental wild type and mutant plants did worse out of the 4 groups of plants, which led us to believe that the BTS gene in the cold has more negative impacts than positive on the Arabidopsis.
This graph shows the bolt height of the Arabidopsis thaliana plants in millimeters over the period of time in which we conducted the experiment. As shown in the graph, only our wild type control and a mutant type control bolted. The wild type experimental and the mutant type experimental did not bolt at all. The wild type control bolted 25 days into the experiment and reached a sizable bolt height of about almost 45 millimeters. Our mutant type control did not bolt until 30 days into the experiment, barely bolting at all, only being about 1 to 2 millimeters. This graph implies that the removal of the BTS gene and effects of the cold have a negative effect on the bolting of the plant.
When side by side, the wild type control and the mutant control plants include several differences. As seen in the image, the wild type control was the only plant that bolted. After 6 weeks of the experiment, the average rosette width for the wild type control was 62.8 millimeters, while the mutant control was 51 millimeters. Unlike the control plants, the wild type experimental and the mutant experimental plants do not include any drastic differences. The wild type experimental's average rosette width at week 6 was 51.9 millimeters, while the mutant experimental plant was 42.8 millimeters. As you can see, neither of these plants bolted. Here's an image of all the plants in group 3. As you can see, the wild type control plant was the only plant with a somewhat successful plant growth and the only plant with a successful plant bolt, while the wild type experimental, mutant control, and mutant experimental plants all had similar results. After 6 weeks of this experiment, our class's hypothesis was disproved. The mutant experimental plant did not grow according to our hypothesis, but not only did the mutant experimental plant not grow according to our hypothesis, but none of the plants in our experiment grew as well as predicted. We believe that there may have been an issue with one or more of the controls in our experiment, An example of this being the light duration of the plants. As a class, we also decided that the cold temperature variable does not have any positive effects on the Arabidopsis thaliana, and in our recent observations, a variable such as pH change on the plant would have a more positive effect on the plant. For future experiments with a cold temperature variable, we believe the plant should be placed in a more controlled environment, so that way it would prevent any issues within the experiment and result in a better plant growth. We would like to thank Mr. McDowell, Virginia Tech, and BRVGS for giving us the opportunity to conduct this research. Is there any questions?
You stated that you placed the plants in that environment of your.
I'm sorry, can you please speak up? I can't hear you.
You stated that you placed the plants in the environment of 50 Celsius degrees Celsius.
Yes, you did.
How did you control the lighting while the experimental plants?
The experimental plants were in a reverse daylight cycle for the lighting. We placed them under a fluorescent glow light from 3:45 p.m. when we took them out of the fridge until 8:30 the next morning when we put them back in.
What was the reasoning for the variable that you chose?
Chose the variable of cold because the Vaz gene is shown to have an effect on how plants withstand different temperatures. So we decided that cold would have as much an effect, if not more, than pH.
So I've got one question. The temperature, I didn't hear it clearly, but it sounds like it was cold treatment was at 50 Fahrenheit. Is that right? Okay. You know, it's possible that those plants might not have actually perceived that as a stressful condition, because Arabidopsis generally is found at northern latitudes and it grows in early spring. So, you know, in that context, it might have perceived 50 degrees as relatively balmy. So you actually might not have stressed them out quite enough in regard to cold. Great presentation.
Dr. McDowell, so we have a question, right? So 12 years of doing this, we've never seen plants, like, well, I've never seen it, where they grow rosettes like this and then they don't fold at all. Like, that just, what could have caused that? So would you guys like? Yeah, you want me to speak for all students? I would too. So somebody's block free to talk about how you did the lights. Quick volunteer.
So the lighting, in the fridge, there was no light, like they were in the dark from 8:30 in the morning to 3:45 in the afternoon. After 3:45, when we took them out, we placed them under a fluorescent glow light with a timer to turn off at 8:30 the next morning before we put them back in the fridge. Yeah, we decided that there may have been an issue with one or more of the controls. The light duration was one of the controls that we thought, but there may have been another issue.
2 and my block 3 were right on top of each other, two different road lines. But my block 2 has great growth, great bulbs, all of them. The block 2, again, different set of road walls, but I don't notice that it's that much. I was kind of perplexed by it also.
Higher temperature on block 2 if they were above block 1.
That is possible because they were window level. Block 3 was at sub window level. So they were hitting a little bit, but not a little bit yet. That would be my hypothesis.
Lights are kind of adding up the total amount of light that they've got. And incorporating temperature into that too. And integrating over the course of their lifespan how much total light they've accumulated at high enough temperatures to actually generate enough energy to produce seeds, right? The combination of light and
I'm Jessica Sherman, Jackson Garnett, Olivia Humphries, Isabel Tusing, Tyler Downs, Sadie Wentz, and Mia Rollins. The purpose of our experiment was to test the effects of drought on mutant and wild-type Arabidopsis thaliana plants. In 2017, 821 million people did not have enough food to eat. It is estimated that in 2050, we will have 9 billion people to eat, yet less resources to do so. Current plant diseases are destroying over 40% of crops. This is a gene in plants that stops the intake of iron. Popeye, on the other hand, activates the intake of iron. In this experiment, we mutated the plant's Brutus gene to not function as efficiently. These mutant plants have been more resistant to diseases. As a result, science is curious as to whether or not these plants will grow better in drought. Drought is when a plant does not receive enough water to maintain a healthy soil moisture. When a plant does not maintain a healthy soil moisture, it fails to carry out its vital growth processes, such as photosynthesis. It also fails to absorb water, and it is more likely to catch disease and
It was hypothesized that the controlled mutant plants would do worse under both experimental conditions and controlled conditions. One main point of emphasis was the difference between the wild-type plant and the mutant plant. The wild-type plant was a common Arabidopsis thaliana, whereas the mutant plant contained a mutation in its Brutus gene. Another key point of emphasis was the watering schedule. The controlled plants received consistent watering, whereas the experimental plants underwent a light drought simulation. This is because previous studies have shown that when a plant reaches 25 grams in weight, it will undergo severe drought. We chose 35 grams because this would more accurately emulate a light drought simulation. A few ways we collected information was by rosette width, bolt height, and seed pods produced. A few ways we eliminated error was by having 5 groups test our hypothesis, as well as having consistent measurements throughout the experiment.
Graph 1 displays the trends of data between all the plants for rosette width. For the the wild type control, you can see that it starts the lowest, however, it goes up the highest and maintains the highest rosette with the rest of the 28 days for phase 1. You can see that it has a spike by day 14, and this is the day when Rob was supposed to administer all the plants, keeping this one the highest growing. You can see that the wild type experimental had a dip in day 11, and this can be due because some data was factored in that had plants start to grow or the plants had died, and that data was factored in. And you can see that after this, the data became more steady and it became the second highest. It ended up with about a 50 millimeter rosette width overall. The two lowest growing were the mutant control and the mutant experimental. But these two had a very similar pattern. In fact, you could practically say they were the same. However, by the end of the experiment, the mutant experimental surpassed the mutant control by a few millimeters by the end of phase 1. A few common trends were observed in our data of the bolt height graph from days 28 to 45 for our experiment. The control plant grew consistent and constantly the slowest throughout the experiment, but it conserved its water and used water the steadiest throughout the experiment. The wild type experimental, represented as pink, grew the tallest bolt height, but on the days we watered, for example, day 35 and day 43, it had increased water usage as a result of its drought conditions. The mutant control and mutant experimental were very similar throughout the graph, and they used around a steady amount of water usage, and they grew about the same. Another way in which we measured data was by the number of seed pods produced by each plant. The number of seed pods that a plant produces is an indicator of its reproductive success. Two main patterns were noticed within the seed pod count. Firstly, both of the plants exposed to drought conditions produce slightly more seed pods than plants in control conditions. And secondly, both of the Brutus mutants produce slightly less seed pods than the wild type plants. Aside from numerical data, qualitative observations were also taken. From the beginning of the experiment, it was evident that there were color differences between wild type and mutant plants. Mutant plants had slightly dark green leaves, whereas the wild type plants had sort of a light green and yellow leaves. After the drought was administered, more trends were noticed in the data. When comparing both of the wild type plants, wild type control was very healthy, but the wild type experimental had very many yellow, brown, and when comparing both of the Brutus mutants, the Brutus experimental had some slightly yellow leaves, and the Brutus control very healthy and a bit shorter than the wild type control. For my comparisons of plants.
From the data shown in our results, it can be seen that the wild type control plants grew the best of all of our tested Arabidopsis plants. These plants had the largest rosette width, and while they might have had one of the smallest bolt heights and one of the smaller seed pod counts, this can be due to them entering the reproductive stage of their life last, which can mean that they were under much less environmental stress conditions than any of the other plants. The plants that undeniably grew the worst were the wild type experimental plants. These plants had the smallest rosette width from the beginning of the experiment, and while they might have had one of the larger bolt heights and larger seed pod quantities, this can be due to them entering their reproductive stage first, which could mean that they had more time to develop these structures and allude to more environmental stress than any of the other plants had endured. The mutant plants that we tested and grew in relatively similar patterns and trends throughout the experiment.
They had an average rosette width and a below average bolt height and seed pod quantity. And their below average seed pod quantity could be due to them having the Brutus gene reduced from their gene pool, which, as we hypothesized, led to a lower amount of seeds shown at the end of the experiment.
The results from this experiment support what was hypothesized about the Brutus mutant and wild type Arabidopsis. Based on the results, it can be concluded that the mutant plants are resistant to drought because the experimental and controlled rosette width and bolt heights were about the same. Although the mutant plants handle stress better than the wild types, the insertion of more DNA keeps them from growing as well as the wild type plants. Whether in perfect conditions or the opposite, which would be drought, the wild type plants grow at a faster rate and reproduce more. For this reason, introducing the plants to drought would have negative effects. It would most likely decrease production.
Since the mutant plants grew a little slower, it would give more room for weather conditions in a real-life agriculture situation. Farmers. Some plants are harvested early for silage, so the nutrient content of the plants would need to be tested in further experiments. Most plants, though, are harvested at the end of their life, so they shrivel up before they're harvested. So height would not be a contributing factor in the usage of the Brutus mutant gene.
Even though the mutant is not the best option for drought, their resistance to other stressful situations could possibly be beneficial to agriculture. In the future, other stressful situations, such as flood or drought, will be tested on the mutant plants in order to determine various solutions for problems. Not only will this benefit the United States, but also other nations where famine and malnutrition are still present.
Do you elaborate on how you simulated drought through the watering process?
We measured the experimental plants, and when they got to below 35 grams, we just with the other control plants, we watered them whenever they need.
So, congrats on manipulating the crowd stress. So that can be a trick. It's very easy to sort of clear the constant mind. So another aspect of your presentation that I really appreciate is how you carry some explicit predictions on that's why we all work on the model plan. Yes, okay, it's easy to do these sorts of experiments that we like to do, but we also always want to have an eye on. So, thanks for commenting.
So there was no like watering schedule with the control plants, or was there? You said like you just watered them when they needed. There's no like schedule with it.
The plants that we had, especially controls, were measured every single day to ensure that they were being watered properly, that they wouldn't simulate any drought at all. So we have two very controlled results from our experiment. Hello, and good morning, everyone. My name is Catherine Young, and this is Samantha Woodward, Zoe Polly, and Joel Croft. Today, we will be presenting to you our 2019 Arabidopsis thaliana lab results from Madison County. The purpose of this lab is to research how plants can control and resist disease and environmental stressors in their environment, precisely what factors cause them to control and resist these diseases and environmental stressors. Some background on Arabidopsis thaliana is that it is a model organism. This means that it is small, easy to grow, and also easy to genetically engineer. Arabidopsis thaliana contains a gene referred to as the British gene. This gene helps in secreting iron and is also seen in every plant, as the plant would not be able to live and survive without this gene. Through extensive research, our class had decided to test potassium chloride on our experimental Arabidopsis thaliana plants. This is because potassium chloride helps in regulating plant metabolism. It also aids in a quality root development. It also helps in water regulation inside and outside of plant cells. This is all overall critical for the amount of stress a plant is able to tolerate in its own environment. After we decided on our variable, we had concluded a hypothesis that if potassium chloride is added to Arabidopsis thaliana, then the growth rate of the wild type will be greater than the mutant type. This is because British gene in the mutant type is less active than it is in the wild type, leading to a more virulent amount of iron set. The class had a weekly procedure of measuring, observing, and taking pictures of our plants. This provided the class with a set of qualitative and quantitative data for our research and our results. And to measure for the rosette diameters and the bolt height. To test potassium chloride on our plants, we had a solution of a combination of distilled water and potassium chloride. This was inserted through a syringe on our plants. The first level of solution we had started off was 12 milliliters of the KCl solution. The experimental plants had received also 12 milliliters of distilled water along with the solution, while the control plants had received just 24 milliliters of straight distilled water the whole experiment. As we had discovered that the 12 milliliter solution was too much for our plants to control, as they had shown wilting and death after this, we had transplanted some of our live mutant type plants with our dead mutant type plants. After this, we had lowered down the solution to 6 milliliters. We had then discovered death and wilting with our plants, so we had transplanted once again with our wild type plants from extra plants from Greene County. After this, we had moved down the solution to 2 milliliters before finally stopping testing the variable because it was not creating a successful environment for our plant to grow in.
In this graph, it shows the average rosette diameter of group 1. In the beginning of the graph, both the wild type and mutant type experimental groups were declining, while the mutant type control group was inclining. On October 1st was the first time we transplanted, and that explains the rapid incline
October 1st and 3rd. On October 3rd, we transplanted using Greene County's extra plants for the second time. And while we thought transplanting a second time would aid in our plants' survival, it did not. Transplanting a second time made the plants decline in health and caused their death on October 9th. And then we did not do any other things to help experimental plants grow any longer. While both of the control groups remained fluctuating throughout the experiment, slowly inclining until October 16th, when they started to decline. We believe that the control groups remained increasing throughout our experiment because we did not add the variable potassium chloride. And we believe that the experimental groups died so quickly because we did add the variable potassium chloride. We had fluctuating temperatures within our classroom and the added stress from transplanting twice. In this graph, it shows the average rosette diameter of group 3. In the beginning of the graph, both of the experimental group plants had grown very quickly, and then they had decreased very quickly. That was because we transplanted, and the same thing that happened in group 1 happened in group 3. And then they declined, while we thought that transplanting a second time would help. Group 3's plants, it did not, and it caused their death, and they also died on October 9th. While both the control groups remained fluctuating throughout the experiment, slowly increasing until the very end of the experiment. We believe that the control groups remained increasing because none of the variable was added, and we believe that the experimental groups died so quickly because our variable potassium chloride was added, and it was fluctuation of temperature within our classroom and within watering. This is both of the rosette diameter graphs side by side for comparison, and there are several trends to be acknowledged. One with the control groups, how they remained increasing until the very end of our experiment, and also with our experimental groups, how they died very quickly, and how transplanting a first time helped them and spiked their growth, while transplanting a second time ultimately killed them to the point of no return. In the graph above, a continuous trend is displayed between our control bolt heights. More specifically displayed in red and blue, the control bolt heights have a rather flush consistency until they have a rapid increase towards the end of our experiment. We believe that the control plants had this rapid increase, that the control plants increased due to their exclusion from the potassium chloride. Our experimental plants received the 12 milliliters of potassium chloride and therefore had a sudden spike and decrease, leading to their ultimate death. Also, we believe that the control plants ultimately died due to their limited exposure to water and overexposure to the potassium chloride. In this graph, it displays a minimal preview of the typical Arabidopsis thaliana bolt heights. As shown, this plant did not bolt until much later regarding the previous bolt heights. More specifically, Group 4's mutant control Arabidopsis thaliana plant did not bolt until 2 7-day weeks after the remaining groups. We believe that this happened due to its overexposure to the potassium chloride and its limited watering. As for the experimental plants, we believe that they did not bolt due to their overexposure, to their limited watering resources and the inconsistent temperatures within our classroom. This is our Arabidopsis thaliana compare graphs between Group 1 and Group 4. As you can tell, in Group 1, all of our plants ultimately bolted at some point in time, while in Group 4, only one plant bolted.
We believe that in group 4, only the control plant bolted due to it not having that potassium chloride injected into its soil. As for group 1, we think that the experimental plants had a sudden increase and then ultimate decrease, which led to their death due to their overexposure to potassium chloride, their limited water, and inconsistent classroom temperatures. For our increased control groups in group 1, we think they lived longer and had a higher result in bolting, being that they did not receive that potassium chloride and had just distilled water, causing them to have a better growth and reproductive chance.
After conducting our experiment, making observations, recording both quantitative and qualitative data, we decided that our hypothesis was incorrect. This is because we had hypothesized that the wild-type plants would grow taller than the mutant if we add the potassium chloride solution to them. But these plants died before reaching a significant bolt height and when we could compare the two. One thing that we did notice in our control group of wild-type and experimental group of plants were purple leaves. This is because those plants were stressed due to the varying temperatures in our classroom. And some days it would be cold and other days they would be warm. And because of the 2 transplants that took place. Another thing we noticed was a grayish substance forming on top of the soil. And after doing some research, we found that this is actually a harmless saprophytic fungus that is letting us know there is some type of problem going on within our plants. This could be overwatering, poor drainage, or even contaminated potting soil. After looking at our data and comparing our graphs, we noticed that the alterations that the mutant plants received in their British gene had varying effects on their growth. One thing that we can conclude from this experiment is that plants that are exposed to increased amounts of potassium chloride do not survive well, and we know that this will not help in increasing production, making it more efficient and more sustainable for the growing population. Are there any questions?
Why did you only show group 1 and 3 for the rosette width and only group 2 and 4 for the full height?
So did you guys research the negative effects of potassium chloride before you did it? Or was it only supposed to be the positive? We had just researched the effects of potassium chloride, and with all the multiple websites we went through, it had given us only positive ways to grow the plants with potassium chloride. Will you please go back to your graph for the rosette width for group? How come the last of experimental plants, their rosette width goes to 0 and then it spikes back up again? Is that because you transplanted them, but is that testing comparable then, since you have 2 different groups of plants?
We transplanted with our mutant type, with our extra mutant types that we had grown the first time, and then the second time we transplanted was with Green's extra plants. We did not test a different variable the second time. We just transplanted our plants in order to have a fair experiment.
All right, and you
Do you guys know the molar concentration of the potassium chloride that you used? Because maybe that's why they all died.
Our molecular concentration was 1.2 of our KCl solution.
All right, we're gonna have to wrap our questions there, because we have lunch here. All right, guys, we got a couple of things. We're gonna make this. You do want lunch, right?